This video will help you get started with sequencing your personal genome and will provide you with background information on how each type of genetic test is performed. There are many different paths you could choose to follow when deciding to obtain information on your own personal genome. While no one path or one method is necessarily better or worse than the other, there are a few considerations you should take into account when choosing the best path for your needs. These considerations include biological and ethical implications of personal genomic information, such as knowing that your genetics are not your destiny, and that the scope of personal genome analysis is still limited by missing heritability and incomplete knowledge of disease biology. It's also important to note that the genome can be affected by your environment. These effects are studied in the field of epigenetics. Another big consideration should take into account how much information you would like to receive about your genome. This will answer which type of test you will be looking at getting, such as a genotype assay, a full exome sequencing, or a full genome sequencing. A genome array is the most easily accessible type of test. It is offered by many direct-to-consumer genetic testing companies, such as 23andMe and Lumagenics, and those that market directly to physicians, such as Navigenics. All of these companies follow similar DNA procurement procedures in which the customer supplies the company with saliva or blood sample collected using the collection kit supplied to the customer through mail. This sample is then sent to a laboratory for processing. During processing, the DNA is extracted from the sample for genotyping and sequencing. This raw or interpreted data is then sent back to the customer. This method offers the customers the lowest resolution view of their genome when compared to the exome or full genome sequencing. But it is a more affordable method, making it the most common customer choice. The most common genotyping method uses DNA microarrays, which are commonly called SNP chips. These measure more than 1 million SNP loci, or single nucleotide polymorphisms, per sample. These SNPs indicate regions in the genome whose sequences vary between individuals. Therefore, using polymorphic SNPs of interest across the genome can capture the bulk of an individual's genetic variation in a genome that is 99.9% identical to the rest of the human population. Direct-to-consumer genotyping tests generally cost about $200 but targeted genotyping methods, such as those that use Taqman seed genotyping assays, can offer customers a more targeted and cost-effective test option. This slide shows a SNP chip that's used in genotyping. SNP microarrays match the DNA in the sample to a set of pre-made probes. These probes are short segments of DNA that contain different versions of a given SNP and are fixed to a small microarray chip the size of a microscope slide. Since different populations and ethnic groups have different linkage structures, SNP arrays are usually designed for a specific target population. After sample DNA is fragmented and processed, it is hybridized to the array where they base pair to the complementary probes. The array is then imaged, and sample locations can be detected and measured by the fluorescence of a region on the chip. These sections fluoresce because the sample DNA fragments were labeled with a fluorescent dye. Full exome sequencing offers a higher resolution measurement of a personal genome than direct-to-consumer genotyping. Exome sequencing only measures the 2% of the genome that contains gene coding regions. So while it is a fraction of the cost of full genome sequencing, it is still more expensive than genotyping. Full exome sequencing costs can range from about $400 to $1,500. And the popular consumer choice platform, 23andMe, has recently initiated a limited pilot of an exome sequencing service. To sequence the exome, genomic DNA is fragmented and the coding regions are captured using labeled DNA probes, which are single-stranded DNA of each exon. This complementary nucleotide sequence detects the presence of the corresponding exon and then hybridizes with their target. The paired sequences are then captured and isolated using magnetic streptavidin beads. Another exome sequencing method involves microarrays, which also separate exons from the whole genome. The collected exons are then sequenced. During sequencing, errors are inherently introduced at various points along the process, particularly during PCR amplification. To mitigate analysis errors resulting from processing errors, the sequencing technology will sequence every region multiple times to generate a high sequencing depth, which means that the regions are at high coverage. For example, a region at 30 times coverage means that each base was sequenced 30 times. Since exome sequencing uses less sequencing regions than full genome sequencing, less sequencing data are required to achieve a similar sequencing depth and accuracy. This is the main component responsible for the lower cost of exome sequencing compared with full genome sequencing, putting the cost of exome sequencing just above that of genotyping arrays. A full genome sequencing would be the best method to obtain a complete measurement of your personal genome, including rare and private variants. The first rapid method for sequencing a DNA fragment was developed by a biochemist named Frederick Sanger in the 1970s. This method, called Sanger sequencing, used PCR amplification to replicate the DNA fragments to be sequenced. A major drawback with this method was that the reaction would prematurely stop, leading to a mixture of fragments. Since then, sequencing methods have been optimized to allow for efficient sequencing of the entire genome, as seen in next-generation genome sequencing. Full genome sequencing starts out similarly to genotyping. A biological sample, such as saliva or blood, is collected and the DNA is isolated and prepared for sequencing. Common sequencing methods use sequencing by synthesis, where complementary DNA strands are generated by DNA polymerase. The addition of each base is observed using imaging techniques. It is at this imaging step that full genome sequencing really differs from genotyping. Where genotyping assays analyze a static image, full genome sequencing arrays are imaged multiple times as nucleotide bases are added during DNA synthesis. These images illustrate the sequencing strategy used by a popular full genome sequencing platform called Illumina. Here, sampled DNA is randomly cut into fragments and attached to adapter sequences. These adapters cap each end of the fragments and allow the DNA to attach to the surface of the array. Several rounds of PCR are performed to amplify the DNA and form clusters of identical fragments. The fragments are then sequenced via short reads, which are generated via imaging strategies. The first base of a fragment is identified using labeled reversible terminators. The process repeats to generate the whole sequence of the fragment. Once a genome is sequenced, additional analysis is performed to determine the presence and form of genetic variants in a process called variant calling. The most common approach compares each position in the sequence sample to the coordinate position in a reference genome. Areas in the sample that differ from the reference have variants. De novo assembly is another method, where the full genome sequence is assembled from reads without guidance from a reference genome. This method completely removes assembly bias that is inherent in the reference-based assembly, therefore potentially reducing errors and identifying variants. The major allele reference genome method uses genomic data generated by the HapMap project and the 1000 Genomes project that measure genetic variation across populations. Another major aspect that must be considered when choosing whether or not to sequence your genome is privacy and storage methods. Oftentimes, genetic information will come in a downloadable file format. These files must be stored safely to avoid cybersecurity threats and data misuse. While these transfers are often regulated and secure for trusted sources, you must be informed on where the personal data is stored to ensure your comfort with the security requirements. This sums up this introduction to genetic tests. The references listed here can be used if you would like a more in-depth explanation of any of the processes discussed in this video. Thank you for listening.